What?
Is it neural mechanism?
Yeah. Okay. So we've covered a whole bunch of stuff in terms of signaling and predation and all that kind of stuff. And but obviously, with animals, almost all animals, we've got some sort of nervous system. It might be as simple as a neural net in like cnidarians, sea anemones, and jellyfish and things like that. But pretty much all animals have a nervous system. So we're going to look a little bit into that and kind of tie it into what we've learned before. So there's innate behaviors and fixed action patterns. They seem simple, and you should probably read the chapter 12 on, I think I put some pages on there. But they seem kind of simple. Obviously, you have a stimulus, and then it kind of usually goes to, it runs its course. But a lot of the behavior is actually somewhat complex, and there's a lot of different things actually going on, especially neurologically. I mean, if you think about it, there's usually some external sense that's bringing in information from the outside world, whether it's visual or olfactory, auditory, touch, whatever it is. And then there's a whole bunch of processing that goes on. And then there's sending out signals for muscles and everything to go ahead and do the behavior. So even though it seems like fixed action patterns are pretty simple, they're not quite so simple. And even though they're generally innate, or at least parts of them are, there's and they can be altered from behavior in terms of learning and modification. So, kind of classical conditioning sort of things. So, and then of course, we've got learning, all different kinds. Got real simple stuff like classical conditioning and operant conditioning. And then we've got stuff that's really flexible, that requires apparently a fairly advanced neural system to do it. And then, of course, you've got neurons and behavior that also are run by hormones. And so there's the hormonal control that's all part of this as well. I've read some stuff, it's pretty amazing stuff about, when your hormones are kind of screwy, there's some people who, let's say their pituitary doesn't secrete certain hormones anymore. And without it, they have to use, synthetics. Or thyroid is another example, where sometimes the thyroid stops working. You have to supplement it with hormones. But people's behavior and other animals' behavior is really influenced in a large part, kind of like with hormones. We don't really think about it too much, but they really affect our lives. So in terms of this kind of schematic that we looked at before, obviously along the XY axis, the x-axis, we've got highly stereotypical, this is fixed action patterns, and on the right, really flexible. And then in terms of innate or learning, on the y-axis, we've got these things where there's innate, no modification through learning, or maybe a little tiny bit sometimes. And then, of course, a lot of learning involved. So here's our bug at the bottom, eating. So every aspect of animal behavior, of course, has basically a neural component to it. So I mentioned some neurons are bringing in information for processing. Some other elements are being processed. So even though let's say with our eyes, we've got a really good visual sense, the processing, well, perhaps what reality is really like is perhaps much different than what we actually see. We see something that, well, we think it's real. We can certainly move around in the world that way. But it would be, let's think about our eyesight in terms of the electromagnetic field, which is really broad, right? We've got radio waves on one side, we've got high gamma radiation, X-rays on the other side. A lot of what our space telescopes look at, or telescopes, they actually look at, let's say, it could be radio waves, it could be infrared, it could be gamma rays or X-rays. And if our eyes were made right, we would probably be able to see that kind of electromagnetic radiation that's all around us, but we don't. We have a system that kind of processes all this information and gives us a pretty reliable picture of what's going on, and we can make our way through the world based on that representation. But really all it is a representation. So, of course on the left, for you guys over here, we've got this zebra finch, and here's the different, bird brain, some of these different areas that are so important for making songs. I guess that's right. At the very beginning of the semester, someone equated music with the songs that birds make. Does anybody remember that? Or did anybody do that in the discussion?
Say that again?
Okay. It was the beginning of the semester. We were talking, someone was mentioning music, human music, and then how it might relate to bird song. Well, one of you guys wrote it. I don't know which one. And then there was a discussion on it, and how it's kind of similar. Well, it's more like an example of convergence, but there was a recent study that just came out in Science, the journal Science, and it turns out that they did it with a particular bird called hermit thrush. Hermit thrushes use the same general scale that you find in a lot of Western music and other music in the world. Not like some off, where it's completely different, the same one. So, the question was, why does our music sound the way it does? Is that just pretty much contingency or chance? These are the sounds that our brain kind of likes, or is there perhaps some underlying business going on there? And with this work with birds and song, it looks like there's some underlying thing about sound, especially if you look at sound in terms of mathematical. You've got all these tones that are ratios of one another, and that's basically the scales. And it turns out a lot of birds use the same kind of scale that we do. Now, the thing is, there's also a lot of different kinds of human music that, well, they use quarter tones and all that. So I'm not really sure how that applies, but it was kind of an interesting article that I read. It was pretty cool. If you want, I can post it. But anyway, so there's inputs, processing, outputs. So we have some clues in terms of how nervous systems actually control behavior, especially from like fixed action patterns like this is. So we've got these stimuli, the red spot, we've got kind of a bird head but no eyes, pretty close to that. No red spot, even though it's a bird head. And then a red pencil. And so obviously the red spot is the stimulus for the releasing of the behavior, which is for the adult to actually regurgitate food that's eaten for the youngsters. And here's a gull doing it. And there's actually a couple of different things going on here. The young bird is responding to the red spot. And then the youngster pecking on that is the stimulus for the adult to feed the youngster. And we looked at, in this case, visual clues, but a lot of times the cues are, they can be simplified and you can still get the same sort of behavior, like the bull with the motorcycle and all that. And bill pecking behavior. And so you've got this innate in the brain releasing mechanism, a fat, that triggers all these feeding behaviors, but there's more to it than that, obviously, if you took a look at it approximately. Because, first of all, there's the visual thing for the youngster, pecking at the red spot. So it's got to see that, and then it's got to initiate this fixed action pattern of pecking, much like human babies sucking, right? It's a pretty innate response. And then for the adults, you've got this whole situation where they're getting the peck, which is the stimulus, and then they have to go through a whole series of mechanisms, most of it controlled by neurons, which is regurgitating food. And that's not simple. There's all sorts of different muscles involved. It's very complex. So, unlike human males who will mate with anything that moves, male insects will even try to mate with inanimate objects. So, actually, that's probably sexist, isn't it? Against guys, but usually we don't really mind that. It's the other way around that people get upset about. But insects do the same sort of thing. Male insects will even try to mate with inanimate objects if they have the right releaser to initiate the fixed action pattern. So, well, yeah, there is Barbie and those plastic dolls. Never mind. Okay. Oops. Okay. This is a really interesting thing. There's so over here is a cluster of blister beetles. And what they do is they put out a pheromone for this solitary bee. It's a male bee, and it mimics, of course, the pheromone that the female puts out. So the bee comes in to find the female, and there's no female there. They all climb on with all these larvae, and he then finds a real female, at which point they all jump off him and infest the nest, and basically eat up all the eggs in the female's nest. So, let's see. Okay, anyway, so this is the old code-breaking business that we talked about earlier, where other animals oftentimes use code to deceive the animals that they're interacting with. And so this is basically an STD and a solitary bee. And these codes and deceptions are all part of the kind of the neural network. It doesn't take a lot of thinking, obviously, because we've seen with an orchid using a pheromone to get males to come along and pollinate, and orchids don't think, from what I understand. So, okay, let me go. Okay, so this is Dobbington's bat catching a large moth.
Can you replay that? Sorry.
what?
Can you replay that? 'Cause you were
Sure.
walking in front of it.
Oh, I'm sorry. Okay, so let's see here. Okay. Let's see here. Okay. So this is actually a fairly simple thing, even though it seems pretty complex. Bats emit these ultrasonic calls. The echoes come back. They use them to avoid obstacles and also to locate prey. And then in this thing for the moths, there's an escape behavior. So the moths have those tympanic membranes, and they detect the bat calls. And depending on how far away the bat is, they either, if it's far away, they kind of fly away from the bat. But if it's actually near, the bat calls actually cause all the neuronal signals to the muscles to discombobulate. And the actual moth goes into this kind of uncontrolled dive. It can go any which way. It's kind of like, there's things where, what was it? Where you can, I think they've done it with dogs, where you can, certain dogs you can snap your fingers and they'll go unconscious, something like that. And that's kind of what happens. You think, well, that's not perhaps the best way to evade a bat, but it is, because when the bat comes along and they get those calls and they go through this cascade, the response is kind of a random action where they could end up anywhere from their original position. So usually they end up diving down, But they could basically end up anywhere around here just by what their muscles do in response to that neuronal cascade. So that's the basic kind of approach that's going on. Now, who's been commenting on the bat stuff on the student forum? Anybody? About bats that, there's some that are not tasty and they're mimicking that,
Oh, yeah.
right?
About the moths and stuff.
The moths,
Right.
right. Well, there's been some really interesting stuff going on with that. I'll put some stuff on Moodle, but basically, they found moths that make the same sound as other moths that are toxic. So they're using ultrasound to sound like a toxic moth. Basically, Batesian mimicry, or if they're also toxic themselves, Müllerian mimicry. But instead of doing it with a nice bright signal, they do it with ultrasound themselves. Now, it turns out there's another thing going on here. The moths, a lot of moths, especially, which ones was it? It'll come back to me. But anyway, they actually interrupt the sound that a bat makes to find out where the moth is and hone in on it. In other words, they jam the moth's signal so that when the moth comes in, when the bat comes in, at the last moment, their echolocation signal is jammed, and the bat won't catch them. So, let's see here. Oh, I know what I want. Okay, so here's actually what it looks like. This is from a paper that was released in, I believe it was in Science, a couple years back. So the moth actually made a signal that interrupts the bat from homing in on this location. And let me show you another one. This is where the membrane has been deactivated.
So is that like hormonal that happened, or
What?
Or is it hormonal, like chemical? Because we're talking about neuronal.
Yeah, so it's neuronal. So what's going on is you've got this information coming in terms of the sensory stuff is the echolocation, ultrasound. And what this is actually doing is it causes the moth to respond that as the signal gets closer and closer, it gets faster and faster. The moth then sends out a signal, a pulse, that interrupts the bat's echolocation. So this is kind of the classic sort of Red Queen hypothesis where you've got antagonistic coevolution going on. this is where it's been silenced. And they caught it. Okay, so let's go back to the other one. Underneath all that is, you can hear it, it's not as loud as the bats, but it's this ultrasound emission from the moths. So this is the kind of the antagonistic coevolutionary arms race that you find between all kinds of antagonistic interactions, whether it's host and pathogens, prey and predators, competition, all that kind of stuff, where one side is trying to get the advantage of another. And like many arms races, as if you, well, you probably don't remember the Cold War. No, that wouldn't be right. No, you weren't alive then. But back then, there was an arms race between the Soviets and us over nuclear weapons. And one of the greatest innovations that we came up with was something called duck and cover. So they, as little kids, they would say, duck and cover, nuclear explosion. We'd duck and cover underneath our desk because that would save us from the bomb. It was a really good innovation, just so you know. That's why I'm here. And it wouldn't have been otherwise if it wasn't for duck and cover. In fact, duck and cover has almost become like an operant condition with me. So when someone yells out duck and cover, I immediately fall down on the floor and duck and cover. So
Duck and cover.
Right. So anyway, you want me to go down? No, I'm not going to go down now. Later, I promise. So this is kind of, we're a little off track, but it's pretty cool stuff. This arms race is going on. Now, what's really cool is, let's see here. Oh, okay, so, so this is a more recent thing, and this bat, like many others, is hunting for insects, and it's here. Let me make it. Okay, so to counter what these moths have done is this bat is actually like a stealth fighter. It uses echolocation and ultrasound in the same way, but it's really quiet, and the moth actually can't hear it until the bat is almost upon it. Kind of like our stealth fighters, which are basically blind to radar. Same sort of thing. So they found out that, in fact, these bats, these moths, they've been one-upped. Now they're at a real disadvantage because they really can't take evasive action until it's really too late. So they get to within about 3 feet before the moth even detects them, and by that time it's just a quarter of a second or whatever, a tenth of a second before they actually get them. But there's actually more going on here. Oh yeah, we also talked a little bit on that thing about how the moths actually use these tympanic membranes to actually talk amongst themselves. So they not only use it, of course, to discombobulate the bat's signal, but to talk among themselves. So it's kind of an exaptation. Forgot about that.
I have a question on the
Yeah.
race. So is it just a bunch of like
Yeah.
In that example, that moth can deflect the
Mhm.
signal.
Yeah,
That's all it is.
sure.
And then, once they have an equal stance with the bat,
Mhm.
the bat will have to mutate something as well, right?
Well, the bat doesn't have to do anything, right? It's random. If in fact there's some bats that actually gain a mutation that it can counteract what the moth has just done, then those bats will actually leave more offspring than the bats that don't have it. So it's not like they have to have a mutation. I mean, I need a mutation so I can fly. I would love that to happen, but it's not going to happen, right? So, perhaps one day it'll be where, there'll be a human being that has a huge mutation and they'll be able to fly after that, but it's highly unlikely, right? So it's not a matter of needing or wanting, it's a matter of whether some individuals actually have a mutation or not.
So with that moth example,
Yeah.
that the one that deflects the bat's
Right.
ultrasound,
Mhm.
like how is that a recent phenomenon or is that?
Well, it's recently discovered, but it's probably been going on for quite a long time. I don't know,
Do you know if they, like wherever caves that they discovered that in,
Mhm.
they find the same thing in other caves that aren't
well,
cave
okay, so,
replicas?
I think they found, they've only discovered that moth adaptation to disrupt bats in one species. But it's highly likely that it's found in other species as well. Because there's these other things that they found in multiple different species, like the stealth thing that we just saw. That was also found in what's known as a fruit bat. In other words, it's a bat, it's a nectar bat, so it's a pollinating bat. Well, it turns out they also eat insects. They didn't really know this, and they do the same sort of stealth technology where they can come in kind of under the radar, so to speak. So whether it's widespread or not, people are looking into this, and we'd probably use a comparative method to see what groups of moths and nocturnal butterflies use it and what don't.
Sorry, last question about
Yeah,
that. So,
That's fine.
do you know if they actually sequence like the DNA for the moth that can disrupt the ultrasound and the one that can't? And then
I don't think they've gotten that far yet. I think they've probably sequenced some of the moth. But as far as homing in on what genes are actually responsible
Right.
and what neurons are responsible, I think it's a little early right now. That's where they're going to get to.
I was wondering if it exists in every moth. Right?
Yeah.
Like if it's just something that's turned on and the ones that can do it.
Right, right. And that's a good question. It's one of those comparative method things, whether it's a whole lineage that has it or it was independently evolved, whatever it was. Yeah. So, now, there's not just this, but let's see. That's what I want.
We've all fantasized about jamming an annoying person's cell phone. Mexican free-tailed bats have figured out how to turn that dream into a reality. They jam each other's bat signals, but not because they're trying to turn down the volume. They're just trying to steal each other's meals. These bats hang out in colonies that can number in the millions, but living with so many relatives can lead to conflict. When the colony swarms out to feed at night, competition is fierce. To get an edge, the bats jam the sensory equipment of nearby buddies. Then they can swoop in and steal the meal for themselves. Echolocation is basically biological sonar. First you emit a sound, then you listen for an echo, which tells you the position and speed of things around you. And just like sonar, bats can all listen in on the same channel, at a frequency mostly above the range of human hearing. This is the sound of a free-tailed bat getting closer and closer to its target. Often a flying insect, While still an orderly sound, like a normal echolocation call, the audio pulses speed up because the bat needs to get more information quickly as it nears its prey. Unfortunately, this feeding buzz also lets nearby hunting bats know that a competitor is closing in on dinner. Using triangulation, Aaron Corcoran and William Connor were able to show that when nearby bats hear the buzz, they swerve from their flight paths and hone in on the hunting bat. As these interfering bats close in, they actually try to disrupt the hunt by emitting a jamming call of their own. These vocalizations swoop up and down like a siren, covering all the frequencies made in the feeding buzz. The jamming call causes the hunting bat to lose track of its prey, and the jamming bat takes the opportunity to snap it up. We don't know whether other types of bats are using jamming to steal food from one another, but now researchers know to look out for this new adaptation in highly competitive feeding environments.
So
That's cool.
now there's really interesting stuff going on. So there's not only antagonistic coevolution or red queen hypothesis where between the moths and the bats where the faster you run, the faster you have to run just to stay up, yeah?
So, are all the bats in the same population?
Yeah. Yeah, so these are all Mexican free-tailed bats from one they call cave in, I think, New Mexico or something.
So all of them are able to jam each other's.
Yes.
So anything get done then? Do they just do it all at once?
Apparently, I mean, I'm sure there's probably polymorphisms in terms of hunting strategy. So there might be some bats that just go off by themselves and try to find their own insects and away from everybody else. But there's a trade-off there. You're spending time looking for insects versus you're in a big group that finds a whole bunch of insects that are already there. So I'm not really sure what they do, but apparently that's what's kind of going on. The same thing that American and Russian and Chinese fighter planes do. When radar is homing in, we've got all these kind of technologies that will basically interrupt the radar system, and so the heat-seeking or the radar homing missile that's going in will explode at the wrong place. Same sort of thing. So that's what they're doing. Basically, just an arms race to beat the opponent. And like the military stuff, it's evolved. You know, each side is kind of building up new systems that are better and better at thwarting the other side. So, anyway, I just thought I'd bring that to your attention because these arms races, you don't usually get a picture of all that's really going on. You say, oh, okay, so the moths have a situation where the signal comes in and they go all limp and wobbly and go off in any random direction, but then other moths are doing other things as well. So it's a lot more complex. So what we're gonna.
How do you know they're jamming the system, not trying to just get their own meal?
Well, they're looking at the bats that make the jamming signal. It's right on top of the echolocation signal. Okay. Now, it doesn't look like it's. I don't know if they've done this yet. They might have. I'd have to look at the paper. Whether they've done the jamming signal and seeing if it has any properties like echolocation. Because it could. I mean, that's one hypothesis. But from what I understand, it doesn't look like that. That jamming signal is basically a jamming signal that kind of covers up the ultrasound for echolocation. So, yeah. It's a good question. So, and it does look like it's an example of kleptoparasitism, right? They come in, some individual bat is coming in, honing in on a moth, and they jam them and they take the meal instead of actually having to do all the hard work themselves initially. Perhaps. Okay. Okay, so here we were kind of beforehand. So this is the actual sensory apparatus for the noctuid moths. The moths that I was looking at, they're different than these, but they're related. So there's a good chance it's actually something that was heritable that's been passed down to all these subsequent species. Okay. So here we've got, now, here's the thoracic ganglia that are connected to the brain, and then these ear, which is along here, with an auditory receptor, and this is basically what it looks like. You have these air sacs, air sac, and it's like a drum. You bang it, you get the sound back. And so you've got these receptor cells that actually, right here, A1 and A2, that are initiated. So they pick up that difference in the ultrasound that causes, in terms of the ear, and then they send that information off to the brain. So this is the bat that actually does the thing where it kind of discombobulates its flying mechanism. So here's this thoracic ganglia we were talking about. We've got the wing, we've got the ear right here, we've got the sensory neuron, these inner neurons that have to do with the thoracic ganglia, and then of course we've got motor neurons that attach to the muscles. So what's actually happening is there's a change in action potential going on in terms of the incoming signal and the neurons that then bring it to the brain. So this change in electrical charge, it's part of a communication, and it's a communication signal between the different neurons. So what it does then, of course, it relays all this information up to the brain, and then the brain processes it very quickly and sends out signals that are pretty much automatic that cause the muscles to not normally work the normal way they do in terms of flight. So that afterwards, after those muscles are sending out all sorts of weird signals, the direction and velocity that the moth is actually going is kind of a random generation, in which the bat doesn't, it's kind of a random walk. The bat really doesn't have any idea of which way they're going. And so perhaps the moth actually escapes, and then everything gets kind of rebooted after that for the moth, if it's still alive. Let's see, what am I thinking here? Yeah.
Is that why some animals faint?
Well, I think so. Like possum, right?
Or humans?
Yeah, or humans.
Or goats?
Yeah, in fact, or goats, exactly. The men who, what was that? The men who, something goats, that movie?
I don't know, but I was talking about there's actual like fainting goats. They would scare them and they.
Oh, that's right, they keel right over. Yeah, yeah. And there's people that are like that too. You know, it's a kind of a rare mutation, but it's perhaps kind of effective, I would imagine. You know, like me, you know, with the, what was it, where, duck and cover, right? Yeah, same sort of thing, exactly. I think we're going to stop here. I've got this thing that I want you guys to do. It's called, what is it? Hawk moth. So we did that kind of before, the thing is we had too many other things that you guys had to do. So I'll have you guys do this now. Let me go to it and take a look. Okay. Okay. Okay, so it's due next week. It's for the whole class. Yeah, the hawk dove thing. And this pretty much takes you through the whole thing. Let's see, did I put up? I guess I didn't. Yeah, so it'll so unfortunately, it relies a little bit on Excel's kind of tool package, statistical tool package, which a lot of you guys can't get to. Yeah. What? No, this is for everybody.
You're not getting out of this one, guys.
No,
Sorry.
everybody. So what this is okay. Let's see. Where the hell is it? Okay, so you might have to actually go into a lab with PCs to actually finish this off, just so you know. What? Yeah, I think so. I'm not.
Oh yeah, we're on the floor. Down the hall.
So where is it?
It's down the hall.
Down the hall? Okay, I guess it's down. Yes, down the hall here.
Wow.
So basically, you want to look at the Excel worksheet. This is win, what you get, injury, what you get if two hawks are fighting, and then this is the ESS in terms of the percentages of individuals who are playing the game of each type. So you're either a hawk or you're a dove, and the fitness of that phenotype is dependent on how many other hawks and doves are around at the time. So, what you're supposed to do, if we actually look at this, is, hold on a second. What you want to do is you want to change the values of the costs and benefits, not too wildly. Okay, and you want to find out if there's an evolutionary stable strategy. Now, there'll be, in this little box here, there'll be differences in fitness depending on the frequency of each behavior that's being played. And what you want to do, after you've basically, you're changing one parameter here, the parameter is winning, the cost of winning, or rather the, let's see, yeah. So you're changing this parameter, and what you want to do is you want to find where the ESS is for this when it comes back to 0. So 0 means there's an ESS here in terms of the percentage of individuals who are playing one strategy versus the other. So if we were to change this, let's say, to 100, we'd see now that differences in fitness between hawks and doves. The average hawk is 41%. Let's see. 41. Oh, no, that's what the actual fitness differences are. Okay? And now, let me go back here. Okay. And you kept the same proportion there. So what you want to do is you actually want to solve for this proportion so that we get back to the difference of 0. Now, to do that, okay. So, let's see.
I don't think you posted this one.
Tools menu. Where the hell is tool? Oh, it's probably data. No. Let's see here. Okay, hold on. So, this is D16. It's not the same as, oh Christ.
No, but and it's
You know your work for December? For December? No, they do it like, okay, they used to do it like 3 weeks out, 2 weeks, and when people on Lindsay, so they just post it like every week.
Here we go. Okay, let's go back here. So, when you get to into data, you go to the tools menu, and you go to what if analysis, and you go to goal seek, and you have to have this on D16. So in other words, right now, there's the difference between playing hawk and dove. The hawk has a higher fitness of 25, and with this, that's with, if we actually go up to the top here, that's with these proportions. So you go this, oh come on. Oh. Sorry. Oh, that's what it is. Okay, got ya. Tools. Okay, by changing, oh, okay. Okay. So, what it did was it found a solution to that, where win was 100, and that's the winning strategy in this case, after you've changed it, is all hawk. Okay, so instead of 2 different strategies, dove doesn't work in this strategy. But let's say we put this down to, let's say 10. And we wanted to do the same sort of things again.
There's instructions, right?
Yeah, there is.
God,
That's
I hope
exactly
so.
right.
God, I
Okay,
hope so.
0, and then,
Very confusing me.
okay. So this one, then you change it, if you went win was 10, it would change it to, hold it,
Still the same.
no, that's not right.
Yeah. I'm bored too. I don't know what he's doing.
This is due next week.
Yeah,
Yeah.
okay, there it is. So, that's kind of what you're doing. Now you're gonna make, if you actually follow, this thing right here, you're gonna be making these tables, and you're gonna make, kind of a percent hawk at ESS. So hawk is 0.375, dove would be 625, something like that. And then you're gonna put it on a graph. You're gonna actually graph it, and let's see. Okay. So you record your data on the worksheet and something like this, and then you change the values, and then do the same thing for the cost of injury. And then this sheet lets you tinker with the cost. Let's see. Well, you guys are going to have to figure it out. How's that? Now, I know I've probably completely confused you, but it's pretty straightforward.
Where are
Okay.
the directions? You're gonna put it up, right?
It's going to be, it's all on a Moodle.
It is?
Yeah.
Where?
Look under today.
It's under the lunch.
Not the last
Okay. Oh, I'll put the Excel sheet right now.
one. Yeah, you did.
Okay, so here's the instruction sheet, and then there's further instruction sheet on the Excel thing as well.
And you're gonna be posting the Excel?
I'll do that right now.
If you scroll down, like whatever you wrote is cut off, so you can't really read everything you wrote. Where it says familiarize yourself with how that Excel sheet works. Make sure.
Right here.
Yeah, you see how that's cut off right there?
Down at the bottom?
Yeah.
Oh,
Can you fix that? Because I can't read the rest. Yeah, you see how
all I did was just this, that's all.
No, but it's still cut off on the side.
You see here, like you can't
Oh,
test.
yeah,
Can you fix that?
that's true.
Are we done with lecture?
Are we done with how?
There, how's that? We're done. Now, if you guys want to meet with me about your project, that's fine.
No.
And otherwise, you can go work on it. So, does everybody know what they're doing?
Yes.
Have
Okay.
you looked at our what we submitted already?
A little bit, but I was kind of under the weather, so,
Oh yeah, that's right.
yeah.
Non-cost related question.
What?
So,
Hey,
hey, sorry. Did you turn the roof thing about?
I turned it in and I put your name on it.
Okay.
Yeah, because I figured you didn't.
Okay.
I was like, let me just add his name. So yeah.
Okay.
Yeah, I don't know if we were supposed to turn it in, like everyone's supposed to turn it in, but I just figured it on.
Okay, okay.
In case you didn't. I have a genetics term on Thursday, so I'm free on Saturday.
Yeah, yeah.
I'm probably free. I don't know. I can check my schedule.
Okay.
90%.
Okay, and just text me.
I think so.
Yeah, we'll go from there.
What about Friday?
Actually,
Friday I'm going to be in Houston.
well, if you had Google
Okay, okay.
Glass, you could do that.
Okay.
Yeah, so we can start Saturday. We're not doing anything.
So we'll do one like by ourselves. I guess.
Yeah, 'cause you
Since
have
you're
to
not
bring
like
things
afraid to
up,
walk on campus or anything,
well,
right?
Yeah, yeah.
So it'll actually
Right.
compare
Okay,
what
but-
you're
That's
looking at.
fine.
I'm not trying to
What
like
you're-
pressure you or anything,
Yeah, you're
but like
No, I know a lot of surgeons.
so we'll both do it this one time so we can both like experience the procedure and like the consistency that we need, and then
We'll just go from there.
Yeah,
And
yeah.
alternate or something.
Okay, so I can probably do it Saturday.
Okay. Well, I'll meet you Saturday where we can finalize how we're going to do it and go on from there.
Yeah. Oh, we had something to do Wednesday, right?
I'm sorry?
We had something to do Wednesday, discussion questions?
Yeah, last Wednesday. Yeah, we did. You weren't here?
No, I was sick.
Okay.
You think I convinced him?
I think you did because he was sick too, and he didn't do any. He didn't review any marriage stuff that was in it. So yeah, you can use the same excuse that he did.
Okay. So Wednesday we had a discussion and we're supposed to turn in like 3 questions. I was sick on Wednesday. I can even like find the documents. I don't have it.
Okay.
Is there any way I can turn it in late?
Yeah.
What should I do? Should I email you?
Send it to me.
Okay, so I'll send it through email. Yeah.
And I'll send it for you.
The first one I didn't do, like the very first discussion. That was
No, I know.
It's not what I was actually saying.
Yeah.
You want me to send you a doctor's letter too, or?
No,
Okay.
I mean, I'm gonna trust you for some reason. This is really good on my part.
Okay.
But that's what I'm gonna do.
Okay. Thank you.
Are you gonna post the grades for the discussion?
And then went to go pee, and I was
I'm gonna start working on it. Okay? Yeah.
and so
Second floor.
I know where you park. Second. Bye, Mom.
See ya.
See ya.
So after that, and then you go more in specifics for the proposal. That's the intro, and then for the proposal, I'll learn about that. I'll do that tomorrow. Okay. I'll do it, and then I'll send it to you next week.
I think it's something else. I think it's something
Oh, no, just means grab. Just means grab. Looks like a knife. Just grab.
Oh my god, really? Poor little Troy. Should at least just like toy name Troy. How about the filter paper? Used to live in Detroit.
What's up?
I wanted to talk to you about our papers.
Sure.
So we did our intro.
Mm-hmm.
This is our intro.
Mm-hmm.
So what I sent you, I kind of combined the ideas.
Right.
And then I added a graph.
Mm-hmm.
So this graph kind of explains like animals in captivity, how they yeah,
Right.
and their reproductive success is decreased compared to animals who have
Yeah. Better out in the wild.
so yeah.
Or perhaps it's increased because they don't have to worry about getting eaten. But anyway, yeah. I understand.
Okay, so that's our intro, and then
When you do an intro, put introduction at the very top, just so you so, just in the future. So if you if you look at that, I gave you
yeah, you did the thing.
about the what was it, exercise 11 about how to write a scientific paper.
Yeah, I looked at that.
Yeah. So follow that. That's all.
This-
Okay.
This is it.
Right. Yep. Yep. Good.
Okay, so
Okay.
yeah, we're gonna follow this method. It's
Yeah.
pretty much
Except you're going to follow more exactly kind of that other paper, that was a review paper on heterostyly. So, but good for now. Let me look at this.
Is this a paper?
Yeah.
That's the pen that
No.
This one? I didn't see the one on Harry's.
No, I know. Yeah, I know there's stuff there, but no, I had a friend.
No, it was a real paper.
So you're gonna follow that method.
Yeah, it's on here.
Okay. So it's like pretty much intro.
I think the same thing
Yeah.
too,
Oh.
though, but I'm not really from here, so. Where do you
What
live?
am I doing? I'm think I'm yeah. Okay.
In the Bay Area. So when they were like driving,
Oh,
like,
That's right here?
yeah.
like I was quoting shit.
Right.
Like, yeah, like when I go to like the mall down there sometimes, or like
Yeah.
Best Buy.
So
To Best Buy 'cause
it's actually in the book, but this is kind of what they did. So this is the whole paper. Yeah.
We did an
Right.
abstract.
Yeah. Well, not for the proposal. Okay. So just for the proposal, just what the what this thing says here.
At the Journal or South of the Journal. I'm going to college here. So when I was like hires, like past years.
No, that one. Right. Exactly. So introduction and then proposal and then the proposal part.
Proposals, let's go more into depth. So we found an article on pandas, so we're gonna start. I have 3 articles.
Okay.
So we're gonna explain parts of our articles and then kind of prove a hypothesis, which is
Okay.
our hypothesis can be disproved or proved based on the
Right.
article.
Okay.
So,
So with a research article, you're going to start looking through the literature and finding instances where it's supported versus where that idea is not supported.
so
Yeah.
one was supported, one refuted.
Well, I would do a bunch of them actually.
Not just three.
No, so you're looking at it's kind of a broad topic, right?
Yeah.
Okay. And I don't want you to look at all captivity and reproduction captivity. So, but certainly, let's say within vertebrates or mammals, let's say, how that changes, because they've been very successful with breeding some mammals and not others. Pandas, at first they were not so successful, and now they're much better. Right?
The article said that they're not.
Oh, they're not?
Yeah,
Okay.
because of diseases and everything.
Oh,
So it goes
okay.
against.
So
Okay.
that's what the article.
Okay, got ya. Good. Yeah.
So,
So, I
maybe
mean,
we can use that.
but I would, you should certainly use that. You should, there's a lot of stuff that people obviously breed in captivity successfully, like you just did with the rabbits, right? I mean, obviously that's easy to do. But usually there's failures because of various things. It could be external factors like diseases or not the right nutrition or whatever it is. Or it can be internal, not getting the right, well, I guess that's external too, not getting the right signals. Or it could be the hormones aren't right.
The hormones, yeah.
Yeah.
That's like that's like on a genetic level, right? Because there's articles on that, but it is so complicated.
Well, I know. I don't necessarily want you. I just want you to say, okay, on a genetic level, this is what this article concluded,
Okay,
right? So you don't have to go through and say, this is how they found out. But at least say, okay, so on a genetic level, they found that these certain genes were not appropriately sensitive or whatever, and so that's why it looks like things fell apart. Or maybe it's just speculation at this point that there's a genetic component that's not happening. Like there's something called genetic imprinting, where one sex or the other, there's kind of this conflict between the males and the females. And one sex or the other, they tag the same allele so that it kind of ruins the other side's anyway. There's different ideas there. Okay?
Wait, the other articles I found, I want you to tell me if you're okay with them. So the first one, I found one on sexual selection. Female songbirds respond to differences in male visual display, which was true of female cowbirds.
Okay.
So those differences. And then the article 2, when brought into captivity, wild animals can adapt to domestication within subsequent generations. So adaptations may decrease fitness in natural conditions.
Okay, and do you have anything from that?
That was mine. He's like, it was really.
For this one, yeah, for the article 2?
I wanted to eat that. He's like,
No, article. Where's is that the
These are
article?
the articles, yeah, they're right here.
What is it about?
It's about like
The whole summary of the article.
Yeah, what,
or like, he used to live in Philly. Now I go visit him. Like,
because it's such a broad topic, it's
I always
confusing.
like put my food like at his house.
No, I know. Okay,
So I just got,
And it's about reintroducing domesticated wild mice to social.
okay. So there's been this long
back, and I was like, oh, what's the food? He's like, either
research program on silver foxes in Siberia. They probably even mentioned that there, but they were the foxes. They were artificially selecting for tameness versus artificially selecting for being vicious. Okay, and so I'll look that up as well. I mean there's a huge amount of literature on that.
Okay.
In fact, I think I just saw something new on that recently. It was started by this Russian guy, and it was only after the Soviet Union fell apart that we actually started seeing the data, and it was pretty amazing. So it's silver fox domestication. And let me get rid of this.
oh, those little red things. Yeah, basket. What was that?
Do you have to use 3 articles now, right?
So we
That to be one. Oh.
Rice. Let's go to rice with saffron.
You probably want to go to Google Scholar, you can't find it, but, no, there it is, okay.
No.
Yeah,
Was mine.
go into that and then it'll show,
The zoo.
it'll probably give you a list of articles that it's actually from.
No. Saffron.
Let's pray.
Yeah, it's rice with saffron.
The Soviet Union and Russia, the breeding project was set up in 1959. Ludmila Trut and Dmitry Belyev. And then,
Look them up, maybe.
what?
They're the authors of the article.
Lots of articles. I mean, this is a long-term,
Oh.
right? And then if you put in his name, Belyev,
That's what I'm saying.
there's a new article that this was out.
Oh,
Um,
on Silver Fox.
Yeah, on something, it was something related to silver fox.
Yeah. Nice. Rice. Smells good. It's mostly fruit.
No, not, just go Believ.
What'd you guys eat?
No, I mean, just not silver fox.
Chicken.
No,
No.
just get rid of fox.
Oh, it's a
New
fruit
stuff.
rice.
Is Russian Fox.
Let's see, I just saw it, where the hell was it?
That much rice comes
Let's
with
see,
it? Or is that like leftover from like different plates?
So,
Like,
you know what Google Scholar is?
plate is
Okay, good, use it. Because it'll get you to, it'll get you to, first,
I have to go to class right now, but okay, I'll send it to you and then we'll talk. So the proposal, how long does it have to be? Just 2 pages or as
Yeah.
long as it? Because it's not my entire paper. Of course, I'm gonna be working on it.
Yeah.
I'm getting more into details. So
yeah, you don't, I just wanna know, give me some background, and I wanna know what you're gonna be exploring.
so just like kind of summarize the articles that I'm using, summarize what
And,
they're about,
yeah.
and then come up with a hypothesis.
Well, they've already got, there's a ton of hypotheses, right? So you don't necessarily have to come up with hypotheses.
Okay, just summarize it.
Yeah, what you're doing is a review. What have people found up to this point?
Do I need a hypothesis for the paper?
No, that's for those guys, okay? For you guys, you're just reviewing what's been found so far, okay? And in some cases, there's a bunch of different hypotheses. Like, if you actually look at the early stuff that these guys did, well, there's this idea of correlated evolution, where when they selected for, let's say, tameness, there's a whole bunch of physical characteristics that changed as well. In other words, you couldn't change one without the other, okay? So there's a lot of evidence, there's a lot of hypotheses for why that happens. But that's, that's kind of more basic stuff.
Basically summarize the
Yeah,
article. And
right, exactly.
Okay, and then I'll just email that to you today.
Yeah, okay, yeah.
We can't add it to the thing.
Yeah, just send it to me. You know, if it takes a little bit extra time, I don't care. Okay? Yeah, okay, good. Okay, good.
Summarize it.
That's why it didn't die, because it's never in the sugar.
Wait, why is it 5.6? Oh, yeah, that's not good, right?
There's 2.
How'd that happen?
Those used to be below those. I know we have this question earlier, but
What is the calibration?
CP0.
Well, do you know if there are paper plates in the beer room?
Paper plates in the what?
Beer room?
I think they are. There might be, also in the, stock, not the stock room downstairs, but the ecology stock room, this floor.
That one's only,
Okay, how many do you need?
-4,
Yeah.
Which is the
Okay, I'll go find out. I know the key, so I'll go get one right now.
Thank you. Whoa. That looks like something we would pour
Wait, is that
like
sure this is the
your
sugar? This is
soup.
Oh yeah.
It's like what says in there. That's sugar.
You're gonna have like
Oh, that's the.
like 15 on
Yeah.
there. Protein.
Is that what you guys were asking? It has like
I don't know, you guys have to look at it.
mixture.
This is child too splendid.
So I wasn't sure.
Charlton
I had bags
sugar.
of that.
Try not to
Why?
sugar.
Put the sugar.
It's only meant.
Did you put the sugar?
Look really gross.
I did.
No, but it was just
It came out
originally
like
6, so it was 0.1.
Yeah.
That was,
That's close enough.
Oh,
because then
so what
one
was
packet
it
is
now?
3.
So they took away a gram?
48 grams.
No, they took away one.
Well, we should measure the sugar.
Should be fine.
So what did we do? How did it become? It was like 5.9 earlier.
Hey, someone take a picture of the sugar. Take a big picture of the sugar.
Oh my god, that's fine.
What do you think of Cheerios?
Oh, how much?
We're doing
No.
How much did the thing weigh? How much did this weigh?
We're making lunch.
worms.
Making lunch. Are you hungry?
A little
We
bit.
got blueberries,
How much
rice,
is
Cheerios,
How much is the
tuna, and
paper?
mealworms.
0.4?
1.4.
So subtract it from 6.1.
Oh, and beans. Oh shit, how is it?
Okay, what, so what was the other one?
I almost
6.1,
forgot,
right?
and I pulled out of my parking structure. And I was like, I don't know what it is. I park no street.
So the sugar is, this is 6.1 earlier, minus 0.4, so 5.7, so they took away 3 grams.
Yeah, so it was just 1 gram. Because it was 5.9 earlier.
So they took away.
Well, let's just take the sugar.
Wait, wait, wait,
Let's
wait.
do the sugar.
Yeah, we could ask them to. I don't know. I like meet the summer, but I don't know if it's.
We said in your last one.
Wait,
Yeah,
You
the
lock it.
beginning, I don't know if there's stuff.
2.8 plus times 2.
Maintenance sugar is 4.
I have the bag candy, but I was waiting for my students. I was just like, okay, leave it here. I'm like, sure. But I forgot to put my keys in the bag until I went to go tap that. It was like 8 o'clock.
So then,
So what'd you do?
I went to the recruitment's room, like, those contacts her office, and he's like, no, you can call
Well, we didn't know it
Pops.
would be that much.
Or you could go to her house.
So
And
how
I'm
much
like,
is it?
neither of those seem like viable options to me.
You can call the cops, like,
How much is it?
Help, help,
How's it
help.
more?
It was 5.9 earlier, now it's 8.9.
what the hell?
It's probably the oil one I have.
Oh,
Wait,
right,
so
you're
I'm
just
gonna
scared.
take it.
That sucks.
They shot you. I don't know. You guys.
Oh, wait, never mind.
I hate when I do stuff like that.
Okay.
6.2 minus the 0.4.
But no, earlier it was 5.9, so it's
6.1.
My brother drove over.
No, it was
to
5.9.
his bare feet. He's like, maybe.
It's 5.7 now.
It couldn't have taken that much.
You guys do it like
Yeah, it was 0.2 then.
I mean, that's all the sugar.
They're not using razor like this.
Splenda.
Did you just squish the head?
Yeah. This is sugar. These two are Splenda. Because this is child too Splenda. Wait, no.
Are you taking notes?
I am. Yeah.
Well, it's not 5.7 because earlier it was 5.9, which is for sure. Because it was
This math's not a lie, right?
No, because Splenda was the one that was lesser, remember? Not sugar. So
No,
you're
but
gonna
this is
make
trial
it.
too.
It's gonna be
Okay.
consistent. It was
No.
the same for the.
Once we record it, we'll see where we are and tweak it a little bit. See where that.
Right.
Wait,
Look what is this?
take the pants out. So this one now, it's 120, so they took away 0.2. What is the dead end?
No, I think it's still leather, right?
So it's squishy.
Weather,
Now we are
location,
outside.
habitat.
1 point? So it's, what was it? It was 2 grams, right? They take away 0.2. This is trial 2,
How
right?
much does it end?
Yeah.
Just kidding.
22. So the sugar.
Did she say related in Celsius?
Yeah. Look, right now you should make it. You can put it on here the sugar. There.
How many did you
Plus 275, right?
Let's
I
go.
don't know what else. She
The
didn't
trial
say
one.
what she
So
wrote.
what do we?
She wrote that and then.
That's why I was saying it was 5.9, so it was only 1 gram.
Oh, and then it didn't make sense that it was just 2 grams.
Yeah, I didn't have special machines, and I didn't have,
I mean,
Electrode and air.
Now we can just do the data.
you just killed it with your
You
hand.
put trial 2 was,
Okay.
1 gram was lost, and then trial 2 is 2 grams.
point one was lost.
Yeah.
So, we originally had 6 grams of sugar, right?
Yeah. Well, I mean, we're just gonna say it was 2
It was
grams
2
and then 0.9, make it 1.9, so it's good.
Habitat, bark, like, what?
Right. So in general, there really is no difference.
It's
I want to say too little bit.
0.1 versus 0.2. Yeah, that's fine. Our hypothesis would be
There's no difference.
You put C6
prediction.
on though? What else do we have to do?
Is this trial 2? Let's take the
Degrees Celsius. Is it cloudy outside?
answer there. Very good. That's moving a little bit.
But you can take it out.
I'll just take it out.
Is it sun out?
Yeah.
Sunny. No, it's
Yeah, it's
cloudy.
It's not.
5.6. Was that slow?
Beautiful.
Much better.
Who's today? Beautiful Tuesday.
So these are, now, these aren't just paper plates. These are special. Ooh.
Fancy.
There's a special to it, just
Uh-huh.
for you, guys.
That's right. I want you to know that, okay?
Well, we
That's
were
how we-
feeding them before Thanksgiving, so
What?
thank you.
Oh, sure, absolutely. Yeah, I guess it is.
5.7.
Oh, that's great, yeah.
Check there before Thanksgiving though.
Yeah, yeah. See,
It's a stretch.
feed them turkey.
Just
I
like
feed them
5.7.
turkey. That's what I
Okay,
would
it's 5 so.
maybe,
Yeah,
that's what I would like.
it's-
No, turkey's much better.
I like ham.
Do you?
Wait, how much
Yeah.
did you say?
I mean, ham gets too rich, you can't
It was
even
like
make it
2.8,
real turkey.
so it was like 5.
What?
My favorite
Yeah.
is chicken.
It's different.
Well,
because another
well,
one was-
but
yeah,
there's
there was
also,
more
you know,
sugar than
like-
the second time.
So that's
Stuffing, what about
so that's
stuffing?
1.7.
Yeah.
Hate cranberries.
You don't like it?
No.
Oh, God. Cranberries.
Oh, this is
great.
You haven't had the red cranberry.
No.
I've never even tried it. I don't even like the way it looks.
Listen, for this Thanksgiving, you go over to her house.
Yeah,
Okay.
vanilla bourbon
Okay.
cranberry sauce.
I
I
don't
just
even like how
invited
it
you.
looks.
Okay, that's
Perfect.
fine. That's fine. You're welcome.
Well, I think I'm already covered. I wish I could be like 3 different people. 'Cause
Me too.
I've got one in the Sierras, I got one down here, 2 down here, it's just a
So we
mess.
do Friendsgiving. Friendsgiving. On a different day.
Yeah.
That way.
Well, it would be not much nicer, I admit it. So,
Do a potluck. I know
yeah,
this.
yeah, no, I could do that too, you know. I can make, I'm pretty good with turkey
You make good turkey?
and stuffing and
Did
all
you
that
deep
stuff.
fry it or you baked it?
No, I was baking it, but, I think, I'd start brining it first. Because brining, it really does seal all the juices. I mean,
You
that's,
should bring one in for the class.
yeah, you're right.
Yeah, we should just have a party.
God, that would be great.
That'd be fantastic.
I make really good green beans and greens.
Uh-huh, good.
Nice, really nice.
Yeah, so we'll see. Yeah, you guys organize it, will you?
You bring the
I'll
turkey.
pour 'em.
Okay. I'll bring the turkey, you guys will get everything else.
Got it.
Yeah.
We
And
haven't
we
had
could even
stuff
do a
figured
deep
out.
fry here, I'm sure, you know, it's like.
I never had deep fried.
Oh,
I
yeah,
heard it's
it's,
amazing.
it's pretty good, yeah. No, it, you actually grind that too, but,
It's more sugar.
yeah, I was surprised. Doesn't take quite as long, but that's part of the whole thing.
Yeah, my aunt starts like the day before Thanksgiving.
Yeah,
We start like the week before.
yeah.
And
How much,
pieces.
what do you have?
Like 15.
No, that's fine. Or is it
So did you put that down?
Mm.
Okay. And then we just throw it away.
And you have three
God,
bars.
I've been,
Yeah.
No, but keep it.
I've been craving Cheerios.
Yeah, I know. We need
What's
to.
up?
Yeah, no, that's okay. I got some at home.
No, those are regular sugar.
Yeah, I think I'm gonna go
We don't
home
use
right
that.
now and get some Cheerios. I'll feel better.
Oh, we do? Table sugar?
Christina, what class you have
So you
today?
don't only have-
Yeah,
Oh, okay.
I've recovered kind of.
So we just need to put that
2 grams.
close down. So we get to.
Yeah, you should go take a nap then.
Okay.
I will.
Or do you want?
But I need to,
We need paper, right?
I need to,
That's why.
record a couple of lectures from the other class I
Or just throw
had.
it back in
That
here.
that they weren't recording.
Throw the sugar.
We should take a nap first, so you like energize to do it, and then wake up and do it.
Or I can
Or
do
do you
it
just wanna put
two
it in
different
one
ways,
now?
an energized one and a sleepy one, and see which one's best,
Yeah,
right?
but then that's double the work for no reason.
Well, it is, but you know, it puts them to sleep.
Exactly, that's why you need the energized one, because if you're sleepy, they're gonna be sleepy
Well, no, I'm
watching it.
thinking that's good, because then otherwise, if they were energized, they'd go out on the street and do crimes.
What class are you teaching, right?
Right?
What class is this?
Oh, Biol 100.
Oh.
Come on, I mean, you know,
Yeah.
it's dangerous, like, it's dangerous people in there. You know, I mean, that's real behavior.
Yeah.
Instead of just studying.
Especially because they're freshmen.
Yes, exactly.
I'm gonna write
Oh, God,
Or no,
right.
it's one.
It's right there.
Okay,
No,
so,
I have a team here.
I'll lock that and then you guys can leave whenever you want. So, as far as the scale, oh, they're doing it.
Can you just
They're
know?
doing the
No,
scale?
we're
Sherry just stole it.
They stole our scale.
They did, okay.
It's me, nice.
So,
And it's good.
anytime you guys need to scale, just let me know and,
Well, he wants us to just leave it back there and he'll pick it up when we're done.
yeah. But the thing is, you might need it a couple different times. I mean, which ones are you putting, what is it, bird feeder stuff?
No, this is, sugars and,
So are we
Oh,
gonna do
okay.
like a big
We're
We're
doing
doing
this.
so far we haven't seen a difference.
I think we should do it more often than that, mostly, because if we would almost every day, we would have to do it. So it's our discussion.
So what's the initial rate this one?
Like,
This is trial?
I would say twice a week. No.
so we would do it twice a week.
Oh, this is trial,
Well, it depends on, so what's the measurement?
The hell's I
How
thinking?
much food is eaten at different places or what?
Let's go outside.
What do they prefer, protein versus low
We
protein?
work hard, guys.
Okay.
And
Did
then
you
we're
guys
going
wanna add
to
another, or that's
Duck
it?
Pond here 3 times, we're going to Balboa Park
Yeah.
3 times, then we're going to
Oh, okay, got it.
So in trial 8, so now we're
So,
using
and then you're measuring how much of each
a
one is
different
getting eaten.
kind of,
Okay, so when you put it,
so what's the way to this
are
one
you
initially?
just looking, so you go out there,
They're
you,
both 2 grams now.
see how much they eat, and
No, we were
then
sure before.
when do you come back and check it? How much they've eaten, or is it just?
I think
We have to-
I think we're gonna measure it until they finish one of the.
Are you guys gonna stay here?
And so we're
I
gonna
guess
see.
you do.
We have to go outside.
Grams
Oh,
per minute,
okay.
we're gonna know the number of grams we start with, and then we're gonna come back and weigh the second plate that they
Uh-huh.
didn't finish eating,
Right,
and then see how much they've eaten.
okay, okay. And you probably wanna get, you might wanna do a
You don't
scan
have any more
sample
candy?
of how many
Do you have
birds
the
are there,
original?
okay? And you
This
might
is,
wanna actually,
This is just everything put together.
There.
randomize it over the course of a day. So, I don't know how many times you're gonna plan on going out there, but, instead of, just when you put it out, you say, okay,
Oh, no, we're doing
then you
two,
go
right?
out, the first time you put it out, you do a scan sample, then you go out another time.
Oh no, this
you
is
do a
trial
scan
9 and this
sample,
is
you
trial
can
10.
Count the number of birds,
Yeah,
right? And that'll be like a covariate,
okay.
okay? Because obviously that's what's
I mean, we only
driving
need to
this, I would think.
record
Would
the
be,
first
one
and
thing
second,
would be
and then
covariation, and then
Yeah.
what you're really interested in is which one they actually prefer,
Mm-hmm.
okay?
How do you keep this and
So
make
then
it safe
the
when
number of
it's
birds
fall
we
out?
covariate,
Yeah,
and so we would do a hand cover for that?
yeah. How do you learn that? I know, you're in the evolution and ecology group, right?
Yeah.
Yeah. Not like YouTube. What are you in?
BA.
Oh,
BA.
yeah. Medicine, right? Yeah. I'm dealing with people who complain a lot. Right? Isn't that
That's
true?
me.
Yeah, they complain. I mean, they say, I'm sick. I know you're sick. Well, shut up about it.
Garbage.
That was the only kid, like, I was like, oh, it's cancer.
People just
Like,
tell that kid it's not the
do you have
garbage.
cancer? Like, yeah, someone told me I have cancer because I have a bump.
You're
Like, my neck.
contaminating
It's
the
like,
blueberries.
okay, and then I'm like, oh, my arm hurts. Oh, my neck hurts. Oh, my foot hurts.
Yeah.
To the point where, like, if I really was hurting, like,
She didn't listen.
yeah, I'm just
I mean, over the weekend, I had a mild case of Ebola, you know. What happened?
How'd you get through that?
I don't
You know
know.
that?
Obviously, I'm just too nasty for it.
But I
They
mean,
didn't like you as a
No, they
Yeah,
said,
yeah.
oh, shit, let's get out of
That's
here.
like what the mayor of New York said. He was like, oh yeah, Ebola would just hate it in New York. Like,
Yeah,
people want to
exactly.
stay here.
Yeah, the New Yorkers are too nasty. They do much better in Indiana.
Yeah.
Indiana, they bring them into their homes, feed them, all
Yeah.
that kind of stuff.
Invite them to stay for dinner.
Yeah. Okay.
We might need your plastic bags.
Really fun.
So this is, I feel like I might drop it or spill it. Because it's not bad.
Just hold it like you're making, like weed.
Roll it.
Why is it? Oh, there's a hole in it.
Experimental error. It's like monster.
Are you guys staying? You guys are leaving, right?
What?
No, I'll talk to
Oh.
you.
Are you gonna leave the door unlocked?
No, I'm gonna
How about the
lock
scale?
it. Do you need to get it in here again?
Yeah, like after
We just want to record it one more time.
Oh, okay, so you're doing it right now.
Well, we can go to Oppenheimer's lab and get a scale.
Good,
Okay, yeah,
Yeah.
thank you.
good.
Use your.
So I think it was 70.6 for the first thing we had. The first thing was 37 point, oh, wait, sorry. Then it to 71.
I think she did it.
Yeah. We need this.
Yeah.
Oh.
Do you want to?
I even gave them nice beans. I mean, it was from 99 cents store, but I picked up the food and no one picked that up.
72. 22.8. Oh, 22.8. I was like, how the hell?
30.2. 30.1. 31.9.
Actually.
32.
2? Okay.
Is that 1 or 2?
That was 1.
33.1.
Close enough. We're not going to be able to get a 0.2 grams of blueberries. Here, let me replace some of those like less than perfect looking ones with some that they might prefer.
1.1 grams.
It said 33.
Yep.
The age just said 31. I was like,
Wait,
this
hold
is
on, let me
33.
replace some of the nasty blueberries with better ones.
Ah, yes.
Because if they prefer, yeah.
You're over. 32.
32.
30.
30.
Mm.
Third. We'll see.
That one, yeah, that's it. That's 30.3.
There we go. Do you want to zero it?
Okay.
Yeah, and that way each time we're just going to shoot for 33.3.
Why don't you just
Yeah,
go instead?
safer. Let's see how much they like Persian rice. I would still.
It'll do that.
This 100 grams is not very far.
I thought it would be more.
Yeah, I thought it would be way more.
Well, we can, today could be like a trial, I guess.
Yeah. Yeah, I definitely thought it was gonna be more.
So like double that amount?
Are you serious?